biochemical oxygen demand. Apparatus and material. Dilution water, water sample, DO bottle, measuring cylinder. Distilled water, DO meter and BOD incubator. Procedure. 20 ml of water sample was measured using measuring cylinder and poured it into a BOD bottle. Then the bottle was filled with dilution water until it reached full capacity and tightly closed by using a glass stopper. Clean the DO probe by using distilled water. The DO meter probe was placed into a sample and let the DO meter record the reading. Record the DO reading. Clean a DO probe by using distilled water and then put the DO probe into a sample and test again. The steps are repeated 2 more times to ensure accuracy and to obtain an average reading. Then clean up the DO meter probe by using distilled water. Take the sample to the BOD incubator. The sample was placed into a BOD incubator with temperature of 20 degrees Celsius for 5 days. After 5 days, take out the water sample from BOD incubator to test it. Clean the DO probe by using distilled water. Place the DO probe into a water sample to test the DO5. Record the DO reading. Take out the DO meter probe and clean it. Then test the DO5 again. The steps are repeated 2 more times to ensure accuracy and to obtain an average reading. Turn off the DO probe and clean the DO probe by using the distilled water. Data analysis. The BOD5 can be calculated by using the BOD5 equation. The calculated BOD5 for this sample is 22.84 mg per liter. The water sample has BOD5 of 22.84 mg per liter, which indicate a very high level of organic pollution. It falls under class 5, meaning the water is not suitable for irrigation, creation or aquatic life without extensive of specialization treatment.
Experiment 2. Chemical oxygen demand. The apparatus and material that is used during this experiment is 550 ml of water sample, 1 ml pipette, pipette tips, 250 ml beaker, test tube rack, low-range COD digestion reagent vials, distilled water, DRB200 reactor, and spectrophotometer. Next is the procedure. Prepare 2 low-range COD digestion reagent vials and place it onto the test tube rack. Before pouring 30 ml of the sample water into the beaker, the 250 ml beaker was rinsed using distilled water. The water sample is poured into the beaker. And then, the pipette tips is attached to the pipette. A volume of 2 ml of the water sample was measured using a pipette and transferred into a low-range COD vial that was held at 45-degree angle directed onto the inner wall of the vial during the transfer. Then, for the blank sample, the step is repeated using 2 ml distilled water. Then, the COD vial was capped and gently evaporated multiple times to mix. Then, both of the sample and blank vials were placed into a DRB200 COD reactor and heated at 150 degrees Celsius for 2 hours. After 2 hours, wait about 20 minutes for the vials to cool to 120 degrees Celsius. Spectrophotometer was set to program number 350 For COD low range, the outside surface of each vial was wiped clean before testing. The blank sample was measured first to calibrate the spectrophotometer. After the initial blank movement, the sample low-range vial was measured and recorded. The steps were repeated 2 more times to ensure accuracy and to obtain an average reading. For data analysis, the chemical oxygen demand in our lake averaged at 26 mg per liter. This likely exceeds the Class 2A limits of 25 mg per liter, which is the standard for the conventional treatment and protection of sensitive aquatic life. Therefore, the lake water requires proper treatment, especially oxidation and biological process, to reduce organic load before use. Experiment 3. Ammoniacal nitrogen. The apparatus and material that were used during this experiment were deionized water, tissue, beaker, measuring cylinder, 10 ml sample cells, serological pipette, and spectrophotometer. Next is the procedure. Firstly, the sample was poured into a beaker. Then, from the beaker, 25 ml of the sample was measured using a measuring cylinder. 3 drops of mineral stabilizer were added to both the sample and the blank, and each was inverted several times to mix. After that, 3 drops of polyvinyl alcohol dispersing agent were added to both the sample and the blank, followed by gentle inversion to mix. Using 1 ml pipette, the Nestle reagent was added to both the sample and the blank. And then the solution were inverted again to ensure proper mixing. The mixture were allowed to rest for 1 minute. Then, each solution was then transferred into a 10 ml square sample cells. The outer surface of the sample cells were wiped clean using a lint-free tissue, and the blank was placed into the spectrophotometer to calibrate the machine. The sample cells were cleaned using lint-free tissue, and the sample was then inserted into the spectrophotometer to obtain the absorbance reading. The steps were repeated 2 more times to ensure accuracy and to obtain an average reading. For data analysis, the ammoniacal nitrogen concentration reading averaged at 5.29 mg per liter. Based on the NWQS, this falls under class 5, which means a very poor quality and not suitable for any use. High ammonia levels like this may indicate contamination from urine, sewage discharge, or fertilizer runoff, which is common in urban lakes. Experiment 4. Total suspended solid. The apparatus and material that were used during this experiment were tissue, 10 ml sample cells, distilled water, 500 ml beaker, blender, and spectrophotometer.
Procedure. A 500 ml water sample was prepared. The sample was poured into a blender and blended for 2 minutes to ensure even suspension of solid. After blending, the sample was poured into a clean 600 ml beaker. A 10 ml portion of the blended sample was measured using a measuring cylinder. The 10 ml portion of the blended sample was transferred into a 10 ml square sample cell. Another 10 ml sample cell was filled with 10 ml of the tap water to serve as a blank. Both sample cells were sealed with stopper. The outer surface of the sample cell were wiped to remove any smudge or moisture. The sample cell containing tap water was inserted into the spectrophotometer to calibrate the machine. The blank was removed and replaced with the sample cell containing the blended sample to obtain the absorbance reading. Step 10 and 11 were repeated 2 more times to ensure accuracy and to obtain an average reading.
Data analysis. The total suspended solid in our sample average at 69.7 mg per liter, which fall under class 3, indicating moderate water quality that requires extensive treatment. This shows that physical treatment steps like sedimentation and filtration are essential before the water enters further treatment stages.
E. coli and coliform test. Apparatus and material. Distill bottle, beaker, colilette, water sample, quantity tray, quantity sealer machine, quantity sealer rubber, spectral line, UV light and incubator. Procedure. 110 ml sample are measured using beaker. Then transfer the water sample into a distill bottle. Add a colilette into a distill bottle. Close the distill bottle tightly and then shake the bottle until the colilette dissolves. After that, the reagent and sample mixture was directly poured into a quantity tray. The smaller well are trapped for 2 or 3 times to release the air bubble. The quantity tray was placed onto a quantity tray rubber. Insert into quantity tray sealer with the well side of quantity tray facing down. The sealed quantity tray was placed in an incubator for 24 hours. After 24 hours, take out the sample from incubator to calculate the amount of coliform and E. coli. Take out the quantity tray sample to determine the E. coli and coliform. The yellow well are counted to determine the coliform. For the E. coli, the spectral line UV light was pointed to the quantity tray and the well with the blue color are counted and tabulated. The amount of coliform and E. coli are determined by using MPN table. Data analysis. The amount of coliform in water sample is 2,419.6 mpn per 100 ml and the amount of E. coli is 1,046.2 mpn per 100 ml. The water sample coliform and E. coli are classified in class 2A and 2B in water quality index. This means the water is contaminated but moderately. It requires conventional treatment before it is safe for use by sensitive aquatic life or recreational use involving body contact. pH experiment. Apparatus and material. pH meter, distilled water, water sample and beaker. Procedure.
A volume of 100 ml of sample water was poured into the beaker. Then, the electrode of pH meter was rinsed with distilled water and directly immersed into the water sample. Once the reading stabilized, the pH value was recorded. Sample readings were repeated 2 more times to obtain 3 readings for averaging.
Data analysis. The water sample has an average pH of 5.85, which indicates slightly acidic condition. It is fall under class 3, which it is moderate quality, but it needs extensive treatment. Turbidity test. Apparatus and material. Water sample, beaker, sample cell, tissue and turbidity meter. Procedure.
80 milliliters of collected water sample was poured into a beaker. From this volume, 10 milliliters was transferred into a clean sample vial. The vial was then sealed securely and its exterior was wiped using tissue paper to remove any moisture or residue. Once the sample was prepared, the turbidity meter was powered on. The vial was carefully placed into the chamber and the read button was pressed to initiate the measurement. The same procedure was repeated 2 additional times to ensure consistency in the results. All 3 turbidity readings were recorded and organized into a table for further analysis.
Data analysis. The average data for turbidity test is 2.5, which is under class 1. It showed that this water is very clear with a low level of suspended solid. It is suitable for conventional use such as supporting a natural aquatic ecosystem. No additional treatment is needed for turbidity.